Hey everybody, Mr. Murphy here. I'm going to talk about 10.4, which is all about translation. So translation is once we have our mRNA and it has exited the nucleus, it's now in the cytoplasm, and it's the process by which it's going to be read and converted into something meaningful, of a signal that tells the ribosome what amino acids to put together into the protein. So there's a whole lot of text in this chapter about the different details of this, but the important thing is that there are tRNAs that are paramount to this whole process. They are the facilitators of delivering the different amino acids to the ribosome based upon what code the ribosome is currently sitting atop. And so while we think about ribosomes are the protein factories of the cell, yes, but the ribosome's sort of like the overseer who's hanging out and saying, calling out for, hey, here's the codon that we've got, and then getting the right TNA to the place. So the ribosome is constructed out of RNA and protein complex. It's actually made out of two pieces that we'll see in a minute. And that is facilitating then a step-by-step process of reading the codons 3 nucleotides at a time. These tRNAs have different functions depending upon their shape and structure, and so I'm going to skip to that. And here we can see that they are a single-stranded RNA. However, that single strand of RNA folds up on itself into a very particular shape, and so it has this sort of shape here, and it's kind of like a reverse Florida. In this diagram, at least. I guess you could look at it from the other side. It has these different hydrogen bonds that are causing that shape. And then there's this little region down here in red, which is the anticodon. And those are going to be the complementary bases for whatever codon this tRNA matches up to. At the top, we have what's called the amino acid attachment site, which is a very creative name for the site at which the amino acid attaches, and this is where the tRNA is going to be holding on again to that amino acid and delivering it to the ribosome. Now, there's a concept called Wobble. You notice that this is a little rounded portion of the tRNA here, and for that reason it doesn't always fit perfectly onto the codon, and that's why for a lot of these different amino acids we see a lack of specificity on that third codon letter. So it gives an example here. Alanine is GCA or GCC or GCU. They all get recognized by the same tRNA. So the same tRNA lands, and as long as it's got that GC part there, that tRNA is going to be landing there and delivering alanine. So there is a little bit of wiggle room for differences in the code, but basically what it means is that GCA, GCC, and GCU don't need to have 3 different tRNAs that match up to them. They're capable of producing alanine as the next amino acid using just a single tRNA. So there's a little bit more to this in that the process of basically recharging the amino acid here on a used up tRNA, it involves ATP, but basically there's a whole secondary or tertiary process happening in the background as tRNAs are dropping off their amino acids. They are then scooping up a new amino acid with the help of some other enzymes. And that's actually what these activating enzymes are. So they swoop in and they help the tRNA to find its appropriate amino acid. Interestingly, a experiment was done where they took tRNA and it had cysteine, and they ripped off that cysteine and stuck on an alanine instead. And basically, they're asking the question: is it the amino acid or the tRNA itself that dictates how it's going to be a part of protein synthesis? And what they concluded was that nope, when we switch out the cysteines for alanine, we get alanines in our protein where we expected cysteines. And so they demonstrated that it is in fact the tRNA, and then subsequently we discovered that it's the anticodon region on the tRNA that is really the key to when that tRNA does its job. But if you were looking for like a fun way to prank your ribosomes, just swap out manually some amino acids. Just an idea. So this is all, of course, leading to the ribosome, and ribosomes have 2 subunits, the large and small. And so the ribosome sort of constructs itself around the mRNA in the cytoplasm. And there's 3 different regions to that ribosome that we'll go over right now. So the regions are the amino acid site, which is where the anticodon of a charged up tRNA with an amino acid, lands at the amino acid site and matches up to its codon that it is complementary to. And then the polypeptide site is where the the next one is coming in, and we're starting to build the amino acids together into the polypeptide. And then the exit site is where the used up now tRNA is going to exit without its amino acid. So it has dropped it off here at the polypeptide site, and now it's leaving at the exit site. And so this, similar to the transcription process, we have initiation, elongation, termination, and very similarly, it's pretty straightforward. The names kind of tell you what's happening. So initiation is where the tRNA and the small ribosomal subunit bind to the mRNA. And then, as I said here, the small subunit moves along the mRNA until it reaches that start codon, and that's when this first amino acid is going to be dropped off. Now, that's always methionine, and I think it's important just to note that is often removed. The question comes up a lot in class where somebody raises their hand and asks a good question, like, does that mean every protein that our body makes starts with methionine? And no, because sometimes that amino acid isn't right for the attractions and things that you want in your folded up protein. So it would kind of be a hindrance if you always had to have that. But it's what is coded by that promoter region when we were transcribing, and so that's sort of the signal to the ribosome, consistent signal that's where we need to begin. So when we here's the picture I was trying to find. When our methionine comes in, that's when our large subunit comes in and says, well, it's time to do our job. And so the ribosome is now whole here and begins with our first amino acid. Subsequently, elongation occurs. I'm just going to skip ahead. I think there's a picture. And this is where the next tRNA is going to come in, bind to the codon that the anticodon is attracted to, and then that's now going to allow the original methionine to get tacked on to this new proline amino acid. And meanwhile, then the tRNA that had dropped off the methionine is now free to go. And then we would get our next one coming in, which has tyrosine. The proline and methionine are going to be handed off to that. The next, the second amino acid carrying tRNA that has now let go of its amino acid is able to go, and then it's just keep building and building and building from there until we reach what's called termination, and this is indicated by a stop codon. And a stop codon, instead of attracting a tRNA, attracts what's called a release factor, which is basically just then bumping into this amino acid chain and the last remaining tRNA and saying, okay, get out of here. And that's when the ribosome then knows, okay, it's done, and the small and large subunits can then separate away from each other. And that is the process of translation. So initiation is all about that promoter DNA, which codes for the AUG, and then termination is that terminator sequence on the DNA, which contains the stop codon. It is possible, and this is the last point of this section, that multiple ribosomes could be working on a single mRNA at a time. And so if we're looking at this, maybe this is the first ribosome that's finishing and reaching the stop codon here, but maybe the next one's just over here, and it's also sliding down the length of this mRNA. Even if ribosomes aren't doing this multiple at the same time, a single mRNA, this mRNA here that has been released, is exactly the same as it was before it found the ribosome. So it's just going to keep existing in the cytoplasm until another ribosome binds to its initiator or promoter site and begins the process again. And so a single copy of mRNA can actually make many, many copies of a protein until that poly A tail is slowly getting chipped away at by different enzymes in the cytoplasm. And once all the bogus stuff at the end, the poly A tail is gone, and it begins to break down actual coding regions, that's going to be sort of the end of that mRNA's life cycle in that, and a ribosome is just going to get stuck on it and not be able to finish producing the protein, and that's therefore going to lead to a stop of its process, a permanent stop. If you have questions on this section, let me know. The next section is all about the modification of proteins that happens after this. Thanks.